Okay, so now we're going to talk about protein synthesis. So as we've talked many times in this chapter so far, we have DNA, which undergoes transcription to make RNA. And then the last thing is going to be translation to make a protein. So before translation occurs, we have one kind of prep step, which is the activation of tRNA. So this is that same structure of tRNA I showed you earlier when I introduced what it was. To activate tRNA means that the amino acyl tRNA synthetase, so this is a enzyme, attaches the amino acid to an acceptor stem. So the activation is the actual attachment of an amino acid. So when you attach, you need to prepare a tRNA by attaching an amino acid. So an enzyme attaches amino acid, and then you form an ester bond between the amino acid and the carboxylate at the C3' position. And then the anti-codon loop pairs with a codon on mRNA. So that's going to be what happens during translation, and you'll see in a moment. So that first step is activation of tRNA. It's just attaching an amino acid. Okay. So we have a picture here, cartoon here of what happens, but let's look at translation. So what is translation? Again, it's the process of going from mRNA to protein. So it's a way to remember this is that it's a change in language translation. We're changing the language of the molecule from a nucleic acid to a protein. So you need a way to translate it. So you need a code, right? That's what those codons are for, to translate between the two. So the first step, mRNA is made in the DNA template. So this is transcription. So we make mRNA. mRNA molecule leaves the nucleus and attaches to a ribosome. So up here, nope, where is it? I think it got cut off. But this pink here is your mRNA strand. I think it goes this way. Yep. So this was the beginning of it, and it goes this way. So your mRNA strand goes and attaches to a ribosome, which is here in the purple and blue. You have that ribosome. So that's the machinery to actually make it. So the code is going into the machine to tell it what to make. In order for this to start, you have to have a start codon, and it needs to interact with the methionine tRNA. So the tRNA that has methionine on it is where it's going to start. So incoming tRNA hydrogen bonds to a complementary sequence on mRNA. So tRNA, let's zoom in. tRNA, so let's look at this one here, this one that has isoleucine on it. tRNA is like a Lego building block, or like, it's just the transport, right? It's transporting this amino acid. So a tRNA with an amino acid at that activation site is going to come in, and it's going to recognize, oh, I match my anticodon here, matches the codon on so the 3-member sequence on the mRNA. So this is going to come kind of like a magnet. It's like, ooh, I matched that one. Come in, and it's going to hydrogen bond here. Then, so that's what happened. That's step number 3. A tRNA with the amino acid on it comes and attaches itself. Okay, step number 4 is chain elongation via translocation. So one amino acid at a time. So this guy comes in, he was new, he comes in and he matches here. Then the ribosome catalyzes a bond formation between this last protein in the chain, or last amino acid in the chain, with the next amino acid. So it makes a bond between here. So you'll see peptide bonds, makes a bond in between these. Then this whole thing, the ribosome will just move over, go bloop, over to the next one, and release this tRNA without the amino acid now. It will be released. That's called translocation. So it's translocating to the next place on the mRNA to do the next amino acid. And that's how it built it, one amino acid at a time. One amino acid comes in via the tRNA. The ribosome bonds them together and then kicks out the tRNA so that there's room for a new one to come in. So translocation, one amino acid at a time, is being built. Lastly, once you hit a stop codon, the chain terminates and the ribosome releases the amino acid chain and the mRNA chain. Okay, and then lastly, at the stop codon, protein is released, and then after the protein is released, it folds into its secondary, tertiary, and quaternary structures. Okay, let's go down. So this is just a summary of what we said just a moment ago. I'm not going to go through this in particulars, but if you'd like a summary, you can go look at this as kind of a cheat sheet. I'd also like to say, I forgot to say this earlier, with the codons, you do not need to write this chart down. Anytime you will need this chart for an exam, I will provide it for you. So you do not need to have this in your notes or memorize this at all.